All right, hi everybody. Today I'm going to be walking us through our experiment with osmosis. So we'll be talking a little bit about diffusion. We'll be talking about how water selectively moves across a membrane. So hopefully you've read the little background information about diffusion. The idea of diffusion couples really nicely with the second law of thermodynamics. So if you've done the lecture already where we talked about energy and enzymes, we know that if you have a bunch of molecules, especially gas molecules, all clustered together in one spot, and then you put them in a big container, that those gas molecules will spread out and fill that container. That spreading out of the molecules to the point where they reach stability or equilibrium, that is in line with the second law of thermodynamics. You're taking something that is non-random, which is the distribution of gas in that container, and then allowing it to distribute randomly and evenly and reach equilibrium. And that is diffusion. I mean, it's the same reason like why like a fart stinks up a room. It's diffusion. Somebody lets one go in the corner, and then within a couple of minutes, people on the other side of the room are like, what? And that doesn't happen. It doesn't require energy or anything for that to happen. It just diffuses. It just spreads out within the room. Oops, I got my lab wet already. That sucks. Okay, so that's diffusion. There's a couple of examples in there about football players drinking seawater and why that's bad. There was something about some lady that was trying to drink a whole bunch of water that didn't have any salt in it and why that was bad. And so what this gets into is this gets into our discussion between hypo, hyper, and isotonic solutions. So we call things by their roots. Hypo, Hypo, typically refers to something that has lacking, that it has less. Like if you're hypoxic, that means you don't have enough oxygen in your bloodstream. So that means you have not enough or too little. Hypo. Hyper, like if you're hyperactive, you have too much energy, you're too active. Hyperthyroidism, you're just skinny, you can't put on weight, your thyroid is too active, your metabolism runs too hot. And then there's iso, and iso just means the same. So I always remember iso is in isometric contraction. So if you're just holding still, right? I'm not contracting my bicep, but watch, I'm going to contract it. Now I'm contracting my bicep, I'm engaging the muscle fibers, but I'm not moving the joint. And so that's isotonic contraction. That means that it's isometric contraction is that it's engaging the muscle, but it's staying in the same place. And so isotonic solutions are ones that have the same amount of salt inside a container as outside a container. And so a good way to think about this is that water is always going to travel to wherever the salt concentration is the highest. So when we talk about membranes, like cell membranes that surround a cell, if you take a cell that has a 1% salt solution inside of it, and then you put it inside of a 5% salt solution, the water inside the cell is going to travel across the cell membrane into the 5% solution until the two salt concentrations reach equilibrium. So you can imagine that if a 1% salt solution is losing water, that means it's slowly gaining percents of salt, right? The proportion of salt to water in that solution is increasing as the water is leaving. And then the solution that the water is traveling to is going to change proportionally as well. As you add more water, the proportion of water to salt is going to increase. And so the percentage of the solution is going to go down until the two solutions reach equilibrium. And so if you have a 1% solution inside of a cell, inside of a water solution that is 5%, when you add the 1% cell to the 5% solution, the two are going to gradually, the 1% is going to slowly increase while the 5% is going to slowly decrease until they reach somewhere in the middle where the same concentration or the same proportion of salt to water is matched from the inside to the outside. So that's basically hypo, hyper, and isotonic ideas. So here's where we get into this. So that first question, would you ever give someone pure water in an IV? The answer is no, that's ridiculous. Why would you do that? If you give somebody pure water in an IV, you're going to push water into their bloodstream. Their bloodstream, or you're going to push water in out, like you're going to what's the word? You're going to dilute their bloodstream so much that the amounts of salt in their bloodstream is going to go down proportionally to the amount of water in their bloodstream. So no, you would never give somebody an IV with pure water. You would want to give somebody an IV with isotonic solution, something that is the same salt concentration as the body is used to seeing inside of its cells and inside of its bloodstream. And so that's why IV bags are going to have saline in them. That's why they have a little bit of salt, is the amount of salt proportionally in the saline bag matches the proportionate amount of salt in your bloodstream and cells. So no, you never give them pure water in an IV. What would happen? You would dilute their salt concentration in their body to the point where they would, they would essentially die, like the lady did, where she drank too much water. It would over dilute it would wipe out their electrolytes. You've heard people of having these like water drinking contests and then dying. It's because you're putting so much water into your system without any salt to match it. And what that's doing is it's diluting all the salt in your body, and you need salt at a Pacific, what are we in, ocean? You need salt in a specific concentration in your body in order for your enzymes to function properly and other things like that. Yeah, that's what would happen. Plants, because they have cell walls, will like to have a little bit of pressure on them, or pressure inside of them. So what solution would they prefer? Well, they would prefer well, let's think about it, right? Would they prefer they want a little pressure in them, not outside of them. So they want pressure kind of pushing the walls out. Well, what would that be? What would that look like? Well, that would mean that they would have to be in a hypotonic solution, meaning that what you would want is you would want water to be trying to move into the cell, right? And if the water is moving into the cell, that means that the cell is going to kind of stretch and expand, and that would put pressure inside the cell pushing out, right? Because if you have more salt inside the cell, less salt outside the cell, then the water outside the cell is going to try to move into the cell to dilute those salt inside the cell to match the salt concentration outside the cell. Holy cow, that's a lot, I know, right? But yeah, that's the idea, is that the plants would want to be in a hypotonic solution that they would prefer. Okay, all right, looks like we're ready for experiment 1. So hopefully you guys got my announcement where you've already deshelled your eggs in vinegar. It only takes 48 hours. So what you want to do is you want to put them in vinegar. The shell is going to dissolve off, and it's going to kind of float and look like flotsam and jetsam on the top of your vinegar. Feel free to kind of gently stir the eggs around, making sure the vinegar coats all of them, and then also to rinse them off like periodically just to get, the crap off of them. And then eventually they'll look like this, and they'll be like squishy little eggs. All right. And so you got I did 4 eggs. You guys can do 2, you can do 3. I did 4 eggs, and I'll show you why when we get to this. All right. So we soaked them in raw vinegar. You don't need to watch the video because it's pretty easy. Just put them in a jar and pour vinegar on top. The calcium carbonate in the eggshell reacts with the vinegar, the acid, to make carbon dioxide, and that gets rid of the eggshell. Don't hard boil your eggs, otherwise they don't work like a cell anymore. And then also be careful because these are raw eggs, and so they're still under a membrane and they will pop. And so that's why I made a few extras just in case. All right, so you rinse off your eggs, you put them in the refrigerator so they don't spoil, and then, or you can start the experiment, which is what I'm doing. All right, take your deshelled egg, blot it dry on a paper towel, weigh your egg, and then pour the Caro syrup in a beaker and place your egg in it. So here's what I'm going to do. I'm not going to put them in these beakers, although I should show you guys what we're going to use today. So I've got this isn't kite string, but it's like very thin string that I can use to tie up my my dialysis tubing. I've got my dialysis tubing. We're going to cut this to about 15 centimeters in our next experiment. I've got 2 beakers. I've got a jar of Caro syrup that I had to buy from the store. I got some paper towels. I got my measuring tape so I can cut the dialysis tubing. I got a little cup of water, just tap water, that I'm going to soak the dialysis tubing in once I cut it for a couple minutes to allow it to kind of loosen up so I can add our solution inside the tubes. Okay, so I've got all that stuff laid out. What we need to do is now take an egg. So what it says to do in the experiment is it says to take 1 egg, put it in Caro syrup, like weigh it before, put it in Caro syrup, weigh it after. and then take that same egg and put it in distilled water. I'm gonna do 2 different eggs, and you guys can do this too. You don't have to do it with just one egg. That way I can run the experiment at the same time. So I'm not waiting 60 minutes and then 60 more minutes and then taking those measurements. I'm only waiting 60 minutes. And then also with experiment 2, when we get to that one, we're gonna soak them in like distilled water. We're gonna soak them, or not distilled water. We're gonna soak them in 1 mole of sucrose and then the other one in pure water. And it sounds like we're gonna be doing that for, I don't know, it doesn't say. Well, we'll deal with that when we get there. I assume it's, oh, there it goes. It's gonna be about 60 minutes. And so what I want to do is I want to make sure that I'm doing almost everything at the same time, so that this experiment's not going to take me 4 hours. It's only going to take me maybe an hour and a half. Okay, so that's where we're at. So first things first, we got to, oh yeah, we also need our scale. The lid of the scale makes a very nice wave out. And so I'm just going to put the lid on top of the scale and then turn it on. I'm going to get rid of this so I can see what I'm doing. Okay, turn the scale on. Now we're going to get out one of our eggs and we're going to blot it dry on a paper towel. So we're going to be very careful with this egg. They're very easy to break. The reason why we're blotting it dry on a paper towel is that water has mass. Water weighs quite a bit, actually. It's pretty dense liquid. And so we want to make sure that we're not weighing any extra water with our egg. Cool. All right. So there's my egg. It's nice and dry. I'm going to very gently set it down on my scale. And my egg weighs 80.7. I should probably get it right in the utensil. Okay. So I'm going to record the egg before syrup mine was 80.7. Yours is probably something similar depending on what size of eggs you're using. All right, and now I'm going to take my syrup, and instead of pouring it in my beaker, because I'm going to want to use these later, I'm just going to pour this in a mason jar and set the egg. So I've got one of these like 16-ounce mason jars, not so big. And then I'm just going to take my Cara syrup, and I'm going to pour it in that jar, and then we're going to set the egg on top of that. So pouring the syrup into the jar. You don't have to use the whole jar of syrup if you don't want. Maybe you can want to make caramels later. You can still make caramels later. And then we're just going to set the egg in the syrup like that. And it's just floating on top, and that's fine. So Alexa, set a timer for 60 minutes.
1 hour, starting now.
There you go. All right, so she'll let me know when I need to take that out and weigh it. All right, for the next one. Okay, so what we're gonna do next, and this is why I have a couple of eggs. It says place the egg in distilled water and wait 60 minutes and then weigh it again. But what I'm gonna do is I'm gonna place it in a mason jar. That way I can use these for the next experiment to run simultaneously with this one. So what we need to do is we need to get the weight of our egg again, and then we need to get another 16-ounce mason jar or something like that. Fill it up with distilled water. So this is important. You can use RO water or you can use distilled water, but don't use filtered tap water because it's still going to have a bunch of particulates in it, like salts and stuff, and that might actually do the opposite of what we want it to do. Okay, having said that, I'm going to pause the video. I'm going to go grab my distilled water and a 16-ounce mason jar. Be right back. Okay, I'm back. I got my jar with distilled water. Actually, I used the RO water. And then we're going to take another one of our eggs. And we're going to blot it dry on a paper towel, being very careful not to break it. Remember, it's just a membrane holding in all of that juicy liquid inside the egg. So you got to be very careful. So I've tapped it dry. I'm going to set it on my scale. This one says 73.6. So I'm going to write that down. Weight of egg before. Where'd that go? Okay, we're gonna go weight of the egg. I don't know why that's in there before water. And that's 73.5 for me. So I'm writing that down in my table too. And I'm gonna take my egg, very gently place it into my distilled water. Boom. Alexa, set another timer for 60 minutes.
Second timer, 1 hour, starting now.
There we go. And then now we can do our dialysis technique. So I have these 2 extra eggs if I need those. And now we're moving on to experiment 2. And so this is where we need to make a 1 molar sucrose solution. So if you want to see how to do that, if you haven't taken chemistry yet, it's really good. Go ahead and watch the video from the lab, the hyperlink from the lab. It's really, really good. She does a great and very succinct job of telling you how to make a 1 molar solution. The thing about 1 molar is the way we do a 1 molar solution is we have to have 1 mole, MOL, which is 6 times 0.22 times 10 to the 23rd molecules of a substance. Well, how do you figure out how much whatever 6 quintillion billion molecules weighs of a solution to mix it into 1 liter? Well, okay, well, we can do that by calculating molecular mass. And so what we can do is we can say, okay, carbon weighs like, what is it? Carbon weighs, so go back and look, I think it's 6, and then hydrogen's 1, and oxygen's like 12 or something grams per mole. So if you have 6.022 times 10 to the 23rd molecules of carbon, then it weighs 6 grams. That many molecules weigh 6 grams. God, it's confusing, I know. Okay, so I'm not going to bore you to death with that stuff. You can watch the video. She does a great job talking about it. But what I'm going to do is, because when we do a molar solution, we dissolve 1 mole into a liter, I'm going to do half a mole into half a liter, because these are not a full liter. And so I'm going to fill up 500, I'm going to do 500 milliliters, which is half of a liter. I'm going to do 500 milliliters of distilled water in here. And then I'm going to weigh out how many grams half a mole of sucrose would be of table sugar. And then I'll mix it in, and that'll be 1 mole solution. So I'm going to get that stuff ready, and then I'll start the video. Okay, so what I did was here's my 2. I took my distilled water and I poured them into close to 500. And then now I'm just using a transfer pipette to get the meniscus down to 500. So I kind of overfilled them a little tiny bit. And then now I'm just very carefully and precisely taking off just enough water so they're both at 500. And then I'm gonna get down and look. Yep, they both look perfect. All right, so there's the exact same amount of water in each of those. One of them is just gonna stay distilled, while the other one is going to get a crap ton of sugar in it. So it's going to get half a mole of sugar, which a mole of sugar, I just went online and looked it up so I didn't have to calculate it. A mole of sugar is 342.3 grams of sucrose. So sucrose is just table sugar. So what I'm going to do is ask my Alexa app, what is half of...
I don't know that.
I haven't asked you a question yet. Alexa, what is 342.3 divided by 2?
342.3 divided by 2 is 171.15.
171.15 grams. So my scale is only going to read up to like 0.1 or 0.2. So I'm going to round up. So 171.2 is how many grams of sucrose, how many grams of table sugar I need to put in here. And so it's going to be quite a bit. So I'm still going to use this weigh boat. And if I have to, I'll do more than one, more than one boat. So there's 11. Dude, we're going to need so much sugar. All right, so screw the weigh boat. What I'm going to do is I'm going to get a bigger cup that I can actually like pour in. So something that might work good is just like a measuring cup. So I'm going to zero that out. Yeah, and then that way I can just pour this in, because I'm only at like 30, 40, 60, 70, 80, 90, 100, 20. Oh, it's gonna be close. It's 151. Oh no, I'm losing it. All right, so I've got 156.7 right now. And I'm gonna add that. So let me write this down so I don't lose track of what I'm doing. 156.7. I'm gonna add that to my beaker. Whew. Okay. And then Alexa, what's 171.2 minus 156.7?
171.2 minus 156.7 is 14.5.
14.5. So that's how much more I need to add. Just 14 and a half. So I'm just going to pour that in until we get to 14. Oh, went over just a little bit. It's okay. I can scoop it out with my spoon. 14.0, 4, 5. Okay, perfect. All right, so we'll add that. All right, now I'm just gonna gently stir this up. Now, if you're having a hard time, this is 1 mole, right? So what I did was I took half a liter and half a mole of sugar and mixed them together. Now this might not dissolve very well, and so the one thing I was wondering is if I should like warm it up in the microwave a little bit, and that might help it dissolve. But I'll worry about that in a second. We'll just stir it up like that. Okay, now we need to get our dialysis tubing set up. All right. That's experiment number 2. Okay, so we're gonna cut dialysis tubing into 2 pieces, 15 centimeters each. So let's get our dialysis tubing. Let's get our paper tape measure. Get our pair of scissors. And then we're gonna measure out just about, it doesn't have to be perfect, about 15 centimeters. And then we'll make a cut. Try not to cut your paper tape measure. Okay, so there's one piece. And then cut your other piece 15 centimeters. 15. Same thing, cut that piece. And like I said, it doesn't have to be perfect, just pretty close to 15. Now we're going to take these 2 pieces and I'm just going to soak them in some water. You know, I mean, it says to soak them for 5 minutes. It's not going to take 5 minutes. So meanwhile, let's keep stirring up our 1 molar solution to make sure that all of the sugar is dissolved, because if it's not, then it's not really a 1 molar solution. So you really want to get all that sugar dissolved. Okay, let's continue reading what we're going to do. So once our dialysis tubes get soft, we're going to kind of roll the ends with our fingers and open up the tubes. So we're going to tie off one end of the tube with our string, and then I'll use like a transfer pipette to put some of this 1 molar solution of sucrose in it, and then I'll tie the other end off. And then I'll add distilled water, the pure distilled water, to the other tube and tie that off. Then we'll weigh the 2 tubes, we'll weigh them out before, and then we're going to switch beakers. So we'll take the sucrose tube and put it in the distilled water, and we'll take the distilled water and put it in the sucrose and see what happens. And then we can answer the questions once we have those. Now, in the lab, it asks you to take some pictures of yourself, right? Weighing your egg. You can take a picture of you with the eggs. You don't have to be weighing them. So if they're in the jars already, go ahead and snap a picture. It's fine. Okay. And then take a picture of you with the dialysis tubing. All right, so let's go ahead and get that all set up. Okay, I think we're ready. So my dialysis tubing is going to come out. I've got 4 strings all about the same length tied. And then we're just going to roll one in, and then you'll see it opens pretty easy once you soaked it for a minute or so. Yeah, and then we're going to tie off one end with the kite string. So I like to twist it first and then take the kite string and wrap it around like a bunch. So it's already twisted, and then the kite string, we're gonna wrap around it a bunch, and then we're gonna tie just a regular overhand knot on the end. Remember which side goes over the top first, so I'm going left over right, and then tying it super tight, and then I'm gonna go right over left, and then finish that off super tight. So there, hopefully that's tight enough that no water is gonna escape. Now this one is the one where I need to put the 1 molar sucrose. Now the good news, if I can get that in there, the good news is that all of my sucrose has dissolved, so I don't have to worry about it being inaccurate. It's definitely 1 molar. And as you can see, it's got kind of this dirty yellow color, and that's because I used pure cane sugar. So refined table sugar, sucrose, is just gonna be white, and it'll make your solution a little bit cloudy, but it won't make it like this color. So don't freak out if yours doesn't look just like mine. As long as you got sugar in there at the right weight, you should be fine. And so if you're following along with me, I'm just using a transfer pipette to transfer over my water, my sucrose. And I want to leave enough room at the top that I can twist it and tie it. And then we're going to weigh these boats, or we're going to weigh these dialysis bags before and after. I'm gonna put one more in, and then I think we should be good. That might even be too much. Yeah, it's a little too much. So I'm gonna just dump a little bit back, not a big deal. Twist it, twist it, twist it, twist it. Same idea. I'm gonna take the kite string, wrap it around the base nice and tight. Over, and over, and over, and over, and over, and over, and over again. And then just doing a regular overhand knot, I'm doing left over right. Tying that super tight, and then doing right over left. So that creates a square knot, which should not loosen up. Okay, so there's my sucrose, one with 1 molar sucrose in it. And we're gonna weigh that in just a second, but I'm just gonna leave it until we get the other one done. This sugar water is super sticky. I'm just gonna rinse my hands off real quick. Okay. All right. Next, dialysis tubing.
Oh, right, come on.
So same thing, roll the ends. Okay. Sorry, my kids are home, it's Saturday. So we're gonna twist one end. Take our string, wrap it around super duper tight. Don't worry, they're almost done. They're just excited they get a draw. Today is actually my 4-year-old's birthday. He just turned 4. So we're going right over left this time on mine. Oops, maybe not. Okay. Nope, I'm gonna go left over right. My kite string is kind of coming unwound. So that one's super tight, and then tie one more overhand the opposite way. Okay. Now we just need to put our distilled water in here. So we'll take our distilled water and fill it up. So this should work out perfect where we'll have a little bit of downtime in between the measurements before and after. But we should have enough space between them that we can conduct the measurements for each one without, the timer running out and being in the middle of one thing and having to jump over and do another. And so hopefully we can do these all at the same time with a great deal of success. All right. The reason I didn't pause it is because you guys are going to have to do this at the same time as me anyway, so you might as well just do it with me. And then if I finish before you, you can just pause me until you're ready. It's a little tricky getting this transfer pipette into the gap on the dialysis tube. That's where I'm running into my problems. It's like threading a needle. Okay. Whew. Now I'm going to twist it up. Twist it, twist it, twist it. You know, in our lab in person, usually we would just like tie the tubes themselves. Okay, so I'm just going to wrap it nice and tight several times. And we'll go left over right. I'll see if I can get right over left. My kite string is coming unwound because it's more like craft string.
Whoa, dude!
It's not really.
You're going to make a rainbow? Okay.
All right, that's pretty good. I'm gonna give it one more loop just for good measure, and then we're going to weigh these out. So if you're not done yet, if you're still tying things up, go ahead and finish that and just pause it. I've got my 2 tubes now, my 2 dialysis tubes, and I'm going to weigh them in my weigh boat. I'm going to rinse my weigh boat off because it's covered in sugar. And there we go. So let's tear that. Oh, it's still a little wet. Okay, what I want to do is I want to take a paper towel and blot these dry. Because I'm going to do the same thing when I remove them from the solution, is I'm going to blot them dry. And I want to make sure that I'm not weighing any extra water on the outside. I'm just weighing what's on the inside. So I'm going to go ahead and tear my scale. This one is, I can't remember which one's which, right? This one is the sucrose. This one is the distilled water. I can tell because they're actually different colors. Thank goodness. I wasn't doing a good job of keeping them separate. All right, so I'm just going to weigh it out. I'm going to try to get it all on the weigh boat so that none of it's hanging off. There we go. And mine is 16 grams exactly. So that's my weight of my distilled water before, so 16.0 grams. And then I'm going to take that and I'm going to drop it into my sucrose container. And then I'm going to say, Alexa, set a timer for 60 minutes.
Third timer, 1 hour, starting now.
See? So she's got it down. Okay, so again, I'm making sure there's no extra water on the outside of my sucrose one. I'm going to go ahead and weigh this one out. This one is 17.1. Okay. All right, and then I'm going to drop that one into my distilled water and say, Alexa, set another timer for 60 minutes.
Fourth timer, 1 hour, starting now.
Okay, so now we can, while we're waiting for that, we can answer these questions towards the end. So you can take another picture of yourself with your experimental setup, and we're going to wait an hour. Okay, so what kind of osmotic solution is the 1 molar sucrose? Well, it would appear like it's hypertonic, right? Because it's going to have more solutes dissolved in it than just pure water. And so I would say it's hypertonic. So let's form a hypothesis and prediction about the direction of water movement in the experiment. Well, okay, if the 1 molar sucrose is hypertonic, meaning it's got a lot of molecules of dissolved stuff in it, then that means that water is going to try to move towards the sugar solution, the 1 molar. That's what you would think. And so for my 1 mole dialysis tubing, so the dialysis tubing with 1 mole of sucrose inside of it, when I put that in the distilled water, I think the distilled water is going to move into the tubing. That's how I would predict it, what should happen. And so it should weigh more. That would be the prediction. So my hypothesis is, let's see. So the 1 molar sucrose, because it has more solutes, will attract water, distilled water. Okay. Prediction. So now I can base my. So remember that hypothesis is just an explanation of what's happening and why, and then the prediction is based. It's what is the prediction is what do I expect my dependent variable to do if my hypothesis is true. So my hypothesis is that sugar water has more solutes in it, and that water will follow the concentration with higher solutes. And so my prediction is, well, let's think about it. What's my dependent variable? What's the measured output of my experiment? Well, it's the weight of each tube, right? So my prediction would be that the dialysis tube with the sucrose inside of it will actually gain weight. The water should move in towards the solutes that are in that tube. And so I'd say my prediction is the dialysis tube with sucrose will gain weight, and the dialysis tube with distilled water will lose weight because the water will leave the dialysis tube in the sucrose solution. So the dialysis tube with just pure water in it, that water, I think, in my hypothesis is right, is going to chase the higher solute that's outside of the tubing and go towards the water and go towards the sugar water outside of it. All right. So we've done that. I'm going to pause the video. You can pause the video, wait 60 minutes, and then we're going to jump right back in, weigh all our stuff out, and do our final analysis. All right, cool. Pausing now. All right, so my first timer just went off. That's for the egg and the Karo syrup. So what I'm going to do is I'm going to take it out of the Karo syrup very, very carefully. And then I'm going to rinse it off. So I take it out. And then under water, I'm just going to very gently rinse it. And then we need to blot it dry. So Ryder's going to be watching this for the rest of it. It's going to blot it dry. Hopefully we don't bust it. And then we need to record the weight of the egg when we're done. So I'm just rolling it very gently in these paper towels. It feels like it's completely dry. Let's set it on our weigh boat. And it says 68.2. Now, look out, look out. I need to get... Before syrup, after syrup was 68.2. Okay, I'm just going to put this back in my jar over here. And then now we have to wait for the timer to go off for the other one, and then we'll weigh that one. So I'm just going to pause it while that happens.
3 minutes left.
Okay, so get ready. My timer's, there it is. Alexa, stop. So that's my timer for this one. That's the distilled water one. And I'm going to dump this down the sink while I do it. That way I can catch the egg on the way out. Now this one, I'm going to be curious as to whether or not it gained or lost any weight because I took it out of the vinegar last night. My wife said it was going to pickle it if I didn't. And so I just put it in water with some salt in the water. So we'll see what happens. So I feel like that's nice and dry. Let's set it on there. Don't touch it. And I get 75.9. So let me write that down. 75.9. Well, it looks like it gained weight, which is what I expected it to do. putting it straight in distilled water. So I'm going to drop that in my jar over there. Okay, so now we just have to wait until the dialysis tubing is done. So I'm going to pause the video until those timers go off. Okay, my timer just went off. My 60-minute timer for my dialysis tubes. And there's the one for the other dialysis tube. So they're close. I mean, they didn't stay in at exactly the same time. Alexa, stop. So I'm going to take one out. I'm going to dry it off as best I can. And let's see. I'm going to start with the one. I'm going to start with the distilled. Which one did I do first? I think I did distilled water in the sugar solution. So I'm going to take out my, this is my distilled water that is inside of this sucrose solution. I'm going to pat that dry. Now this was originally 16 grams. So I'm going to make sure I get all the water off the edges. This one was originally 16 grams. Let's see what it weighs now. All right. This one weighs 11.3. 11.3 grams. Okay. And then let's take out my 1 molar sucrose. Holy cow, that thing got full, huh? All right, I would say this one gained weight because it's like so hot right now. I feel like it's gonna pop if it gets any more water in it. Which is kind of interesting. That's something to think about too, right? If you don't leave enough volume, enough wrinkles in your dialysis tubing, then it's gonna limit how much water can actually move into it. I mean, look at this sucker. It looks like a sausage. Let's see how much it weighs. So this one weighs 22.7. 22.7 grams. All right, so now we're at the end of our lab, so we can finish our calculations. See, so when we go to submit this, the one thing we still have to answer is after the 60 minutes, we pat our dialysis tubings dry, we calculate our change in mass, and then was our hypothesis supported or rejected, why or why not? We also have to talk about the egg as well. So let's do this. Let's start with the egg. And I'm going to go to the egg and corn syrup first, and then we'll do that calculation. We'll answer those questions. Okay, so it says change of weight in egg and corn syrup. So it started at 80.7, and mine ended at 68.2. So that means there was a drop or a change of like almost minus 12, but I'll figure out how much it is by asking Alexa. Alexa, what's 68.2 minus 80.7?
68.2 minus 80.7 is minus 12.5.
Minus 12.5. Okay, so the egg that I soaked in corn syrup lost 12.5 grams. Why does the egg weigh less? Well, corn syrup must be hypertonic, meaning it's got more solutes in it. So the water inside the egg was moving towards the corn syrup to try to dilute it down so that there was equilibrium between the solute concentration in the egg and the solute concentration in the corn syrup. So the water was moving out. So corn syrup must be hypertonic. What type of solution is the syrup? Oh, I already said that. So why does the egg weigh less? Corn syrup must be hypertonic. So I'm going to change my answer to the first one. And I'm just going to move that answer down to the second one. Why does the egg weigh less? Water left the egg and moved toward the corn syrup. And what type of solution must the syrup be? Well, then it has to be hypertonic. It has to have extra solute, so the water gives the water a reason to move. Okay, so that answers those two questions. Now, in table 2, remember, I used 2 different eggs. I didn't use the same egg twice. And so I had to change the table because table 2, hey, bud, that's too loud, bud. I had to change the table. Stand by. All right, so if you remember, I used 2 different eggs. I didn't use the same egg. I didn't take the egg from the cornstarch and put it in distilled water. Although if I had, I probably would have seen a greater increase in the volume of water inside the egg, or the mass of water inside the egg. And so what I did was I changed the table where it says weight of egg after syrup, and I just said weight of egg before water, before I put it in water. And I weighted it 73.5. The weight of the egg after I put it in distilled water is 75.9. So Alexa, what's 75.9 minus 73.5? I think I stumped her. I can figure this one out. So it's going to be 2.4. So minus 2.4 grams. It lost 2.4 grams, right? Yes. Yes, it did. Okay, so based on the change in weight recorded in table 2, what kind of solution is the water in relation to the egg? Well, the water has to be hypotonic. Hypotonic. So why do I know that? Well, I know that the water moved from outside the egg to inside the egg, which meant that the solute concentration inside the egg was greater. So the water was moving from outside the egg to inside the egg to try to balance it out. What do you think would happen to the weight of the egg if we did this experiment in reverse? What if we first placed it in distilled water before the corn syrup? What do you think would happen? Well, that's a good question. So it really just depends on vinegar. If vinegar has more solutes than pure distilled water, then when you put it in pure distilled water, it would gain weight. And then if you moved it from pure distilled water to corn syrup, it would lose even more weight. So it's kind of a tricky question. I would think that it would gain weight first. Gain weight first in the water, then lose more weight, even more weight when placed in the corn syrup. Okay, moving on to the dialysis tubing. Okay. All right, so in the dialysis tubing, it says calculate the change in mass. So let's do that real quick. So we take the after minus before. Alexa, what's 22.7 minus 17.1?
22.7 minus 17.1 is 5.6.
Okay, so I gained 5.6 grams in the one that had sucrose solution that I placed into the distilled water. That one gained 5.6. Alexa, what's 11.3 minus 16.0?
11.3 minus 16 is minus 4.7.
So let's see, my prediction was that the dialysis tube with the sucrose in it would gain weight, which it did. The dialysis tube with distilled water would lose weight. Why? Well, let's think about it. The dialysis tube with the sucrose inside of it had more solutes inside the dialysis tube than inside the distilled water that it was soaking in. So the distilled water moved across the membrane into the dialysis tubing, which made it gain weight. Conversely, on the other hand, the dialysis tube with the distilled water in it that we put in the sucrose had more solute outside of it, and so the water inside of the dialysis tubing moved towards the sucrose solution outside. So it lost weight. So was our hypothesis supported or rejected? I would say it was supported because our predictions were correct. And that's it. And so basically just take a picture of this lab, take a picture of the pages that you put tables on and that you wrote questions down on. Now you can do it digitally. You can answer them digitally, like type them in and stuff, and I'm totally fine with that. Just download it as a Word document so that when you upload it, I can actually open it in Canvas. I don't have to download it to my computer, find a program to open it and such like that. So if you want to do it that way, you want to make a digital copy and then download it as a docx, that's totally cool too. You definitely need a picture of you with your eggs and you with your dialysis tubing. So I really only need 2 pictures, just one with the eggs, one with your dialysis tubing, and then also just your lab, whatever tables you filled out and stuff like that. Yeah, just make sure you have your tables, make sure you have your photos. Yeah, that should be it. All right, guys, had a good time. We'll see you for the next lab.